Alright guys, so the next, following on from mutations, we next talk about gene regulation. So it's still to do with the DNA and the expression of genes, but different, it doesn't kind of flow on in a linear way. It's a slightly different topic. We're going to talk about, I'm not going to talk about now whether there's an error in the gene or not, but we're going to talk about how we can vary the amount of the protein that's produced from a gene. So there's two aspects to this. I mean, you know, the way it's kind of set out is an example of gene regulation is the Lac Operon in bacteria, right? Simple organism, simple regulation process. But then I guess the way that that idea is then extended and then applied is in the context of the role of homeobox genes and how homeobox genes themselves are regulators of gene expression in the developing embryo that allows the development of a variety of organisms. Okay, so we'll first talk about this, and then once we have this idea, just kind of, we're confident, we're happy with that, very simple, quite basic, but once we're happy with that, we use that idea to appreciate how a much more complex system might work, still working on these kind of rules. Okay, so we'll discuss this first, and then we'll move on to that. Okay, so here we have a very simple bacteria or bacterial cell, E. coli, and what we're saying is that we need to appreciate that, you know, E. coli is alive because it can use nutrients that it is surrounded by, you know, to build its biological molecules, to use in respiration, glycolysis, to make ATP. All right, and that should not be a new idea. All right, so, okay. So we've got glucose outside, and I guess that's the preferred substrate because you don't need to modify it in any way. Once it gets inside the cell, it can go, you know, into glycolysis, and the bacteria can produce ATP. Right. But problem is, what if you don't have glucose? E. coli can also use lactose, all right? And remembering that lactose is a disaccharide of glucose and galactose, okay? So shall we just kind of simplify that like this? Glucose and galactose makes up lactose, disaccharide joined by glycosidic bond, right? E. coli can also use this if it is around. So it has the genes to metabolize glucose, it has genes to metabolize lactose. So if lactose is present, what it needs, in addition to what it already has, are two genes that it needs to express. It needs, first of all, it needs a protein that will sit in the membrane called lactose permease. Lactose permease, and that allows lactose to enter the bacterial cell. So now it's inside. Okay, lactose is then inside, but it can't simply just metabolize lactose directly. It needs to break the lactose down. It needs to hydrolyze that glycosidic bond to glucose and galactose. Then it can metabolize those monosaccharides which will then enter the glycolytic pathway. Okay? So, what it needs to do that is express an enzyme called beta-galactosidase. Okay? So, this enzyme, beta-galactosidase. And what that enzyme does is it hydrolyzes the lactose into glucose and galactose, and glucose and galactose can then be metabolized to produce ATP. So, there we have it, okay? So, what the problem is that it doesn't express beta-galactosidase and it doesn't express lactose permease all the time, because it would be a waste of energy to build, synthesize, join the monomers of these amino acids, produce the mRNA needed to code for those. It takes energy, it takes ATP, it takes resources such as the nucleotides used to make the mRNA, such as the amino acids needed to make those proteins. So it's a waste of materials, it's a waste of energy to constantly make these two genes when lactose is not present. So what we have is a very simple gene regulatory mechanism which represses the expression of those two genes when lactose is not around. And when lactose is present, that is when those genes are switched on, the repression is lifted, and the genes get expressed, beta-galactosidase is made, lactose permease is made, lactose can enter the cell, beta-galactosidase hydrolyzes the glycosidic bond, and glucose and galactose can be metabolized to produce ATP, and everyone's happy. Okay, so how does this mechanism work? Okay, so how do these genes get switched on and off? Right, so let's now look at the DNA of the E. coli cell. If we looked at the DNA, if we zoomed in really close, what we would see is just a sequence of, or just a chain of sugars and phosphates and sugars and phosphates, and we'd see the different structures of the nucleotide, the nitrogenous bases. But all those, there's bits of those, because of their sequence, they have a very specific role to play, okay? And I'm sure you've seen these diagrams before, but just know that ultimately it's just a sequence of bases. Anyway, there's a sequence of bases, and that particular sequence, because of the sequence of G's, A's, T's, and C's, is known as the regulatory gene, okay? And it's usually given the symbol of I. Don't know why. Okay, I. Okay, further along, in my diagram, I'm just going to, for space-saving reasons, just going to show it next to, but this inhibitory gene could be anywhere. Further along then, let's just say something like that. Further along, there is a region called the promoter region, and we'll give that the symbol P. There is a region called the operator region, and following that, there are some genes, let's just call that Y and Z. Okay? Y and Z, Z and Y. Okay, now, the promoter operator and these two genes, Z and Y, the combination of these genes is called the Lac Operon, because it's these set of genes that result in the production, or the lack of production, of beta-galactosidase and lactose permease. So let's look at this situation. We've got lots of glucose present. We've got lots of glucose present, and so we don't need to express beta-galactosidase and lactose permease. Z and Y are those genes, okay? Z and Y genes code for the production of beta-galactosidase and lactose permease. Okay. The promoter and operator have different roles, and we'll come to that later. So, in the situation where there's lots of glucose, what is going on? The I gene, the regulatory gene, is transcribed, so we produce a little mRNA, and the ribosomes in the bacteria, they read the mRNA and produce a protein, and that protein is called the repressor protein. Okay, repressor protein. Now, the role of that repressor protein is to repress the production of these Z and Y genes. I should have mentioned that Z and Y are called structural genes. They're called structural genes then because they actually code for polypeptides, proteins, P and O. Even though they are genes, they don't actually code for a protein. Okay? You'll see what they do. So, that the regulatory gene is transcribed, it's translated, and when it's translated, that polypeptide chain folds up to form the repressor protein. Now, that repressor protein, what it does is it binds to the operator region. Okay? So the repressor protein binds to this operator region, and what it does is it occupies that, but it also overlaps a little bit on the promoter region. Now, it's the promoter region that binds RNA polymerase. So if RNA polymerase, so let's just represent that, RNA, a bit bigger. If RNA polymerase can't bind this promoter, it won't transcribe the genes. It won't make the mRNA that's needed for Z and Y to be expressed. And so, as long as we have this repressor protein binding this operator region, overlapping part of the promoter region, RNA polymerase can't bind. Z and Y are therefore repressed. Their expression is repressed. So while we've got lots of glucose around, this is what the situation is. We can't transcribe genes Z and Y, and therefore we don't get any beta-galactosidase produced, we don't get any lactose permease produced, and therefore we're not wasting any energy and materials in order to make those genes when lactose is not around. Now let's look at the situation when lactose becomes available, right? So let's say glucose is no longer present. We've got a bit of a problem now because the glucose is not going into the glycolytic pathway and we're not producing ATP. We're in a bit of trouble, but we do have lactose around, okay? So if lactose is present, some of it, even though, you know, the membrane isn't really permeable to lactose, some of it does get through. And if some of it gets through, it makes its way into the cell and it makes its way into this system. Now what lactose does is, it binds this repressor. Lactose can bind to the repressor protein. And when that happens, the shape of the repressor protein changes so that it can't bind the operator region anymore. So let's just show that that is happening. Okay, so that's my operator region right there. But when lactose is present, my repressor protein doesn't have the right shape anymore to bind to the operator region. And because lactose is doing that, RNA polymerase is now no longer obstructed from binding the promoter region. So RNA polymerase can bind the promoter, it can move along and make the mRNA of the structural genes. Okay? And once that mRNA is made, it will be read by ribosomes, and those ribosomes will synthesize the polypeptide chains, and the polypeptide chains will then fold up into their respective tertiary structures and produce the functional proteins. The lactose permease will make its way into the membrane, where it will allow much more lactose to enter the cell. Okay? And there will also be beta-galactosidase that will be breaking that lactose down, hydrolyzing them into glucose and galactose. Glucose and galactose will then be present in the cell, and they can enter and be metabolized into the metabolic pathways. Then eventually that will lead to ATP production. Okay, so that's looking a bit messy, but I hope you followed that. When there's lactose around, a little bit of it will make it into the cell. Not a lot, not enough for the cell to generate a lot of ATP, but enough to relieve the repression of the repressor protein, which is a product of the regulatory gene. Okay, so lactose binds to the repressor protein. The repressor protein can't bind the operator region. RNA polymerase can then bind to the promoter, transcribe the genes, structural genes, into mRNA. mRNA can then be read by the ribosomes to produce the protein, lactose permease, as well as beta-galactosidase, and beta-galactosidase can then break down the lactose, which is now able to enter the cell, into glucose and galactose, and glucose and galactose can then be metabolized to produce ATP. Yeah? So this is a situation where the amounts of genes that are present in the cell are responding essentially to some kind of signal in the environment. And that's essentially what this example is there to show us, is that genes aren't, we can't just think anymore of genes just being there and continually being transcribed and translated. It's much more complicated than that. Genes are, sometimes they're expressed and they're not expressed according to the needs of the cell. And this situation then can be, or this mechanism can then be used by more complicated organisms to switch on some genes in some cells, switch off some genes in other cells, and thereby create different cells. Cells that then might be able to be contractile like muscle cells, or cells that can pass action potential like neurons. So this mechanism, even though it's kind of, the example is given in a bacterial cell, it's there to show you that genes can be regulated. And if you can understand how this could happen, you can understand how, muscle cells have switched off the gene for hemoglobin. And potentially then how, red blood cells have red blood cells had the gene for hemoglobin switched on at some point. Is this making sense? Yeah? So it's giving you the idea, it's giving you the basics of how genes can be regulated, which is then going to be important for you to understand the next idea, which is about differentiation, about specialization and development, how a ball of cells that are essentially all the same can end up being different things in a developed organism. Okay guys, so the next thing we're going to look at is homeobox genes. Essentially, they have a role in making sure that an embryo, which is a collection of cells that are not very different from each other, how do they develop into different cells, tissues, organs? How do they always end up turning into the right thing? How does that happen? How is that regulated? Okay. So essentially, we're talking about a process of differentiation. How do the cells here always turn into the head? How do these cells always end up forming the midsection, the thorax of a fly, for example? And how do these cells form that the abdomen? Now, that is a process of differentiation, but how, what is the factor that lays out this plan? And that factor is the homeobox genes. Now, we're really building now on what we did last time on the Lac Operon, the idea that some external factor can cause genes to be on or off. That's the important idea that we're going to bring to this. Okay? Okay, so I guess we should look at how homeobox genes were discovered, because what we have is an idea, right? So we have an idea that in these cells, all the head forming genes are switched on. And all other genes, very simplistically, switched off. This section, we're saying that all the genes needed to become the thorax are switched on. So, thorax genes on. And in this section, what we're saying that in all the cells that are in this area, we are going to have the abdomen genes on, all other genes switched off. Okay, so how does this happen? What does that? Yeah, so in the thorax genes, say we're talking about legs. So all the genes that will be needed to make a leg will be switched on in these cells. And we're simplifying here. There's obviously more complex things going on because the legs only form there and so on. But we'll have genes for wings switched on in some of these cells. We'll have the genes that will tell some of these cells that you're going to form the main body section, and so on. Okay, and in the head, these genes won't be on, right? Yeah, and again in the abdomen, these genes won't be on in these cells. So what is making sure that that is happening correctly? What is laying out that body plan? Okay, so the definition of the homeobox gene is that these are genes that are laying out the body plan. So let's just make that point there. Body plan, i.e. cells in this area will become this. Cells in that area will become that. Cells in this area will become something else, and so on. And eventually, if everything happens according to plan, we'll get a functioning organism with, everything in the right place, differentiated, specialized. As well as the body plan, it's also making sure that the polarity of the organism is correct, because it's no use having a head in the wrong place, and it's no use having wings in the wrong place. So we need polarity to do that, so that everything is in the right order, orientation. Okay? So this is what homeobox genes do, but what we're going to look at now is what was the kind of sequence of events that led to their discovery. And then through that process, we'll understand what they do. Okay? So we're looking again at mutations, okay? So one, mutations. Mutations such as the antennapedia mutation, where if this is a normal fly head, there were certain mutations that always led to flies, instead of having antenna on their head, they would have developed kind of legs in their place. So instead of these antenna, they would have legs. Kind of gross. Yeah? And there were other types of mutations that led to other types of defects. And it was always that, you know, things would be developed, but they would be in the wrong place. So we're not talking about a mutation in the wing gene that leads to an abnormal wing. We're talking about the wing being fully developed, but ending up in the wrong place. That's an important difference. So these mutations, when all these mutations were kind of looked at in terms of where they were in the genome and in which genes they were, or which genes they were affecting, it was found that the mutations were always in genes that had similar sequences. And the bit that was similar was a 180 base pair kind of section that led, when the genes would eventually be transcribed and then translated, it led to a 60 amino acid region, okay, in the protein. So these genes led to these genes that were always mutated when something dodgy was going on in the developmental plan of the flies. When all these genes that were mutated were compared, they had something in common, and that common thing was a 180 base pair section. So let's just draw that protein out. Okay? So let's just say that this was the protein. This was one of the proteins, and all the proteins that were altered, all the genes that were mutated, always gave rise to a protein that had a similar structure. And when the sequences of these different genes were compared, they had a 180 base pair section, which was highly similar, very similar. And this 180 base pair section in the protein, 60 amino acids that were important. These 180 base pairs that led to 60 amino acid sequence was this, and they called this 60 amino acid sequence the homeobox. So all these genes that were mutated that led to weird developmental issues in flies, they all had this 180 base pair section in their gene that led to a 60 amino acid portion of a polypeptide chain called the homeobox. Okay? And further analysis showed that the homeobox was actually a region of a protein that was responsible for binding DNA. So these proteins, these genes, encoded proteins that were binding DNA in much the same way that that repressor protein binds in the Lac Operon, in the way that repressor protein binds the DNA and stops certain genes from being transcribed. These homeobox containing genes were also DNA binding proteins. So they were binding DNA and doing something. And this kind of agreed, matched, correlated with the idea that they were somehow involved in regulating gene transcription, because obviously, when they were mutated, certain genes weren't being turned on where and when they should be, which was resulting in weird developmental defects. Okay? So this is how it was kind of shown that the homeobox genes, which is what they were called then because they all had this homeobox region, the homeobox genes were involved in laying out the body plan. It was also known that they were involved in binding DNA, and this is how they were carrying out their role. Okay, so this was important. There was another interesting study, experiment done where you had a mouse which had a mutation in one of its genes, one of its homeobox genes, that caused it not to have fully developed eyes. Now, when the homeobox gene of the fly was inserted into the genome of that mouse, when that protein was expressed in those mouse embryos, that otherwise their own copy of that HOX gene was not functional, it led not to the formation of Drosophila eyes, but it led to the formation in the mouse of normal mouse eyes. So that tells you something important, that first of all, that all these genes are doing, all these homeobox genes are doing is switching on or off other genes. Okay? So, even if you take the gene for the HOX, the homeobox genes of Drosophila that are responsible for Drosophila eye development, and you put it in a mouse whose own copy of that gene is not functional, it doesn't form the Drosophila eye in the mouse. It leads to the formation of normal mouse eyes in the mouse. So what do homeobox genes do? They simply switch the existing genes in that organism on or off. Okay? So the mouse that could not form eyes, it had the genes for, all the genes needed for normal eye development were there. Nothing wrong with them. It simply didn't have the gene that would switch those genes on. Okay? So that's important, that homeobox genes are not genes that themselves code for legs, wings, body parts, anything. All they do is they bind DNA and activate or repress the transcription of other genes that themselves will then go on to be important for the formation of eyes, legs, head, wings, and so on. Okay? So in that sense, they are master regulators because the genes for legs, and this is very simplistic, all these genes that themselves are physically responsible for forming the legs, wings, body, etc. The homeobox genes are genes that switch them on in a very kind of similar way to how in the Lac Operon, the repressor protein switched off certain genes. There are other genes. There are other proteins that can switch them on as well. So homeobox genes are therefore gene regulators. They're gene regulator proteins. They can bind DNA and therefore possibly recruit RNA polymerase so that RNA polymerase will then start transcribing genes. Or they might be responsible for making sure that these genes are not turned on. Yeah? Okay? And what we're saying is that here there'll be head homeobox genes on. Yeah? And in the thorax there will be different homeobox genes which will be themselves responsible to turn on all the thorax genes, So we're talking about special homeobox genes that will only be expressed in the thorax. okay, and so on. And the abdomen will again express different homeobox genes. So different genes turned on in different homeobox genes turned on in different places. Okay, yeah, and that is that. The next question is, how do we even get to the situation where we've got different homeobox genes turned on in different parts of this organism? Okay, now just remember, we had a situation where in the presence of lactose we had certain genes on, and when it was absent, those genes were switched off. Okay, I don't know if you can see that. Can you? No. Okay, so what occurs in this situation is that there could be a range of a certain chemical. So there's chemicals, substances in this embryo which don't have the same concentration in the whole organism. And it might be, and in fact it is, that as long as you've got a high concentration of that signal over here, we'll turn the head homeobox genes on. But if you've got a slightly lower concentration of that signal in the embryo, it might mean that those head homeobox genes don't turn on here, but thorax homeobox genes turn on. And as that chemical diffuses down here, we'll have very low concentration here, but in that case, we'll have the abdomen homeobox genes turned on. Okay, so this is how you've got to think of it. Yeah. Okay, so in one particular organism, we've got this situation going on. The other thing that tends to come up is why is it that homeobox genes are so closely related in different organisms? So, you know, if you look at homeobox genes in a variety of different organisms, they are very similar. And when that is the case, it's that those proteins have such an important role in the normal development of an organism that any mutation, any change in those genes results in that organism not developing properly. And if that organism doesn't develop properly, it will not survive, it will not be able to compete with the other members of its population, and therefore it will not survive and pass on its genes with that mutation. So the more highly conserved the gene is, that means the more important it is, because it can't tolerate even minor changes to its sequence. Because even minor changes mean that it doesn't do its job properly, and therefore it has big consequences for the organism. Some other genes can have more variation. For example, let's say eye color. Okay? Eye color, you can have lots of variations because it doesn't have that much of an influence on, you know, the survival of the organism. But genes that are more important in the survival of the organism, they will be more highly conserved, and you'll find that there's less variation even among different species in their sequence, because any change leads to them not working, and if they don't work, that organism doesn't survive. So any organism that did develop a mutation in these genes has not survived to pass on its genes, and so those genes are not around anymore. Okay, so that's one thing. The other thing is about the number of homeobox genes. The number of genes is usually increasing with the complexity of the organism. So if you've got organisms with a very kind of very basic organisms that don't have a lot of different tissues and organs and different arrangement of that, or not very complex, they'll have a few Hox genes or fewer Homeobox genes. Please don't say Hox genes. They'll have fewer Homeobox genes, but as organisms get more complicated, more complex, they then have more Homeobox genes. And it's the way you have to think of it is that it was the duplication of these genes by random errors. It's the duplication of these genes that allowed the organisms to become more complex. Yeah, it wasn't you shouldn't think of it as organisms became more complex and so they needed more Homeobox genes. It was that the duplication of Homeobox genes by the mutation process that we saw before, the duplication of those genes caused organisms to become more complex. Okay, that's about that. So what I want you to remember is that Homeobox genes are transcription factors. They can switch genes on and off. Different Homeobox genes will be active in different parts of the developing embryo, and they will do different things in terms of switching genes, certain genes on and off. Okay, so different types of these Homeobox genes are described. Please just remember briefly what they do, but ultimately remember how they were discovered and the significance of what their job is. Remember what they do and remember how they are thought to do it. On reflection, I think this is a little bit messy, so what I'm going to do is just go through what the kind of current ideas are about how Homeobox genes work, so that all this information will be kind of brought together in one kind of idea. Okay. So bringing together our ideas, because I know this idea about Homeobox genes is a little bit kind of diffuse. So I'm going to try and make it make sense. So what we said was that in a developing embryo, all these cells are essentially identical. They're there because the zygote divided by mitosis many times. All these cells essentially are identical. They have the same genetic information. So how do we get to a situation where different cells are expressing different genes? What we can assume is that in any situation, everything is not always identical. Okay? And what we're saying is that in the embryo, even though all the cells are essentially the same to start off with, they might be experiencing differences in extracellular signals. So maybe there's some kind of chemical in this embryo that happens to be just randomly higher in concentration in one area, lower in concentration in the other area. Similar to this idea that lactose in the E. coli cell, the presence of lactose can switch genes on. Absence of lactose, those genes are off. In the same way, this signal that the cells are exposed to, which is higher in one area, lower in another, could result in some genes being switched on here, other genes being switched on here, other genes being switched on here. And what we're saying is that those genes that are initially sensitive to differences in these initial signals are Homeobox genes. The Homeobox genes so Homeobox gene 1 is on here because there's a high concentration of the signal. Homeobox gene 2 is on here because there's a kind of intermediate level of the signal, and Homeobox 3 gene is on here. Okay, so we have these genes, and each of these genes has a DNA binding region. Remember, they're a family of molecules. They're different, but they all have this DNA binding region in common. However, they're different genes. They will bind to different parts of the DNA. So this region has a slightly different shape. It's complementary to a slightly different part of the DNA. It will recognize a different genetic sequence. And because of that, shall we just give them different shapes? Because of that, they all have a DNA binding region, but because they all bind to different parts of the DNA, they'll bind to different parts of the DNA and activate the transcription of different genes. So, in these cells, this homeobox protein will bind to the DNA that contains all the genes needed for head parts. Okay? Or it will turn on the genes in these cells that will eventually lead them to be a head. Okay? Head part genes, being simplistic here. Okay? Head part genes activated. Okay? And similarly, you know, we also could have a situation where other homeobox genes are also responsible for making sure that the cells in here don't eventually turn on the leg genes. So we have to have a combination of activating head genes and repressing leg genes, abdomen genes, wing genes. Making sense? Right? And homeobox gene 2 in these cells present here will bind to a wholly different segment of DNA, but this segment of DNA has all the genes for thorax, wings, legs, you know, midsection. All those genes need to be turned on here. And homeobox gene 3, which is activated when there's a low concentration of the signal, binding to a totally different part of the genome. Remember, these genes are present everywhere. These genes are present everywhere. These genes are present everywhere, but because the homeobox, they're only being activated here. These genes only activated there. These genes only activated there because of our homeobox genes, protein. Okay, and genes for abdomen. Okay, so I hope that we're happy now. These cells will become something else, not because, you know, they only have the genes, but they only have the genes switched on here. Other genes are there in these cells, but they're not switched on, and that's our situation. All of our cells, most of them, have all of our genes. My eye cells have all the genes to be a neuron, but they just switched off. Okay, and that's likely thanks to these homeobox genes. All right, so these are therefore the master. These are therefore the master regulators. They switch other genes on, and when those genes come on, then those cells become something else. And so, because these cells have the genes for the thorax switched on, they will eventually form thorax, slightly dodgy wings there. Okay, and because that section has got the gene switched on for becoming the abdomen, it will form the abdomen, and there's the head, well, I forgot the legs here. Okay. Okay, and the head, these cells have the right gene switched on, so they will form the head. Okay? So that, guys, is that. Yep. Okay, any questions? Leave me a message. I will respond as soon as I can. Good luck.